The genomic characterization of classical Hodgkin lymphoma has historically been very challenging, and that's because of the rarity of the Reed-Sternberg cells within a Hodgkin lymphoma biopsy. So the malignant Reed-Sternberg cells represent less than 1% of the tumor tissue. And because of this, on standard sequencing platforms, you're unable to sequence the Reed-Sternberg cells, and therefore we know far less about the molecular characterization of classical Hodgkin lymphoma than we do for all of the other lymphoma subtypes. So to overcome this, our group has optimized a flow sorting algorithm to isolate the Reed-Sternberg cells. And we had initially reported on the first whole exome sequencing of Hodgkin and Reed-Sternberg cells, finding alterations in beta-2 microglobulin, which had not been previously described before. And since then, other groups have also isolated Reed-Sternberg cells and performed whole exome sequencing, as well as characterization of the genome of the Reed-Sternberg cell through ctDNA. However, there had been previously no whole genome sequencing of Hodgkin lymphoma, and so our group was interested in characterizing the full Reed-Sternberg cell genome. And this will give us information about mutational signatures and structural variants, which we otherwise would not have. And so we designed a consortium to collect fresh Hodgkin lymphoma biopsies and collected a total of 65 cases for which we performed whole exome sequencing, and 25 of those for which we had sufficient nucleic acids for whole genome sequencing, and recently reported out the first whole genome characterization of classic Hodgkin lymphoma. So I'll summarize just a few of the key findings from that recent study. First of all, we found some significant differences across age groups. So there were two age groups that we identified. First are the kind of the younger adolescent and young adult population, what we would describe as pediatric and AYA patients. This group of patients in our cohort was, I think, between age 7 and 22, and then an older adult group, age 55 and above. And this correlates with the two epidemiologic peaks for classic Hodgkin lymphoma. But one of the surprising things that we found is that the overall mutational burden was actually higher in the younger patients. And so genome-wide, we saw a higher number of molecular alterations in the younger patients than we did in the older patients. And if you look at the mutational signatures that were contributing to alterations in these groups, there's a clock-like signature that was actually accelerated in these younger patients. So you're seeing an accelerated rate of mutagenesis and a higher mutational burden in the younger patients than we were in the older patients. And this was quite striking. We also did a driver discovery analysis and identified some driver alterations that hadn't been previously described before in classic Hodgkin lymphoma. And then lastly, we performed a molecular timing analysis where we tried to deconstruct the sequence of events that lead to malignant transformation of ultimately a B cell to a Reed-Sternberg cell. So this was kind of the first description of that kind of molecular timing, where we actually found in some cases RAG-mediated structural variants, implying that for those cases, the first hit actually occurs prior to entry into the germinal center in a naive B cell within the bone marrow. And then within that, we were able to time the sequence of acquisition of early drivers, intermediate drivers, and then late events such as APOBAC mutational activity and chromosomal gains.